In this module, we will see the replication of the DNA molecule. As we have previously seen the microstructure and minute structure of the DNA, so in this module we will see how DNA is replicated. Before going into the detail of the replication, we must know some of the basic concepts of the DNA molecule, as we have studied earlier, and we know that the DNA molecule is composed of two backbone molecule structures rather. It consists of one of the phosphate group and the pentose sugar, and similar structure on the other side of the backbone, sugar group and the phosphate molecule. The backbone structure has specific orientation, as we see here. Here we can see 5 prime in this point and the 3 prime in the opposite direction. But when we see the other side of the backbone, we see here 3 prime and in the opposite direction 5 prime. So 5 prime here, 3 prime here. It means that this strand is moving in this direction and this strand is moving in this direction. So these two strands are opposite to each other. So the first main step of the DNA replication starts when this DNA molecule is separated into two strands. These strands are separated apart. So this is the first major step in the start of the DNA replication. So here we can see the details, the process of the DNA replication. And after these strands are separated apart from the double helix, the process of the replication starts. So in this process, the main function that play a special protein that is called the helicase enzyme. So these helicase enzyme, they unwind the DNA molecule into two separate strands. After separation of strands, single strand binding proteins, here we can see these are proteins, they bind to the single stranded parts of the DNA. They bind to the DNA to prevent these DNA to be joined together and form the double helix. So double helix is prevented by the binding of these proteins. The important feature of the DNA replication is this orientation, because we have seen in the last slide that orientation is important. Here we can see again, this is the light blue is the template strand, the strand which is separated from the double helix structure. And here again we can see in the other strand, this is the template. So this template we can see, this is the 3 prime and this is the 5 prime end. But in the other strand we can see here, this is the 5 prime and the other is the 3 prime end. So DNA replication always takes place in the 5 prime to 3 prime direction. So it means that in this strand the replication will be from this direction to this direction. But in this strand, the opposite strand, because this is the 5 prime end and this is the 3 prime end, so the DNA replication always takes place from 5 prime to 3 prime. So in this strand the replication direction will be from here to here. So once the DNA is separated into single strands, there is another phenomena after separation. There is a RNA primer that will be added here by RNA primase. And once primer is added to DNA, the DNA polymerase will act on these RNA primers and will start replicating the strand into the double strand. So that is the reason, that's why in this strand we can see these are the small fragments that are replicated, but in this strand we see it's a continuous fragment that is being replicated by DNA polymerase. So the reason is, in this strand there are small fragments because whenever these two strands are separated by these helicase enzyme, the replication has to be started from this direction to this direction. But in this case it will be the continuous process. So that is why the small fragments, they are being generated in this strand. And further, this strand is called leading strand because it is a continuation of this strand, but in this strand there are small fragments, they are called lagging strand. So afterwards there is another enzyme called DNA ligase. It acts to seal these gaps. So these gaps are sealed together, and then after sealing of these gaps the structure will be the continuous one. And further is that the small structures, they are also called Okazaki fragments because they are small fragments, and these are named after the scientists who had invented that in this strand there are small fragments. So this is the whole process in which we can see that the many enzymes, they are involved in this RNA replication. So we have to remember that first the DNA helicase will unwind the double strand into the single strand, then RNA primers will be added through RNA primase, then another enzyme will act on these RNA primers, that is DNA polymerase, and it will replicate to form the double stranded, and lastly in the lagging strand these enzymes, DNA ligase, will act to join the seals. So we have seen in this module that the process of DNA replication is a very complex process, and in this process we have seen that many enzymes are involved: DNA helicase, RNA primase, DNA ligase, and DNA polymerase. So with the combination of these enzymes, the process of DNA replication is completed.